Welcome back. In the last web lecture, we reviewed the structure of DNA molecules and discussed in more detail how their particular structure made them ideal for the function of being the storehouse of information to be passed on through time. So the transmission of information through time, through the generations, through the process of DNA replication. In this web lecture, we're going to begin to think about the other function of DNA as being the instruction book for the building and maintenance and running of living organisms. And this is through holding the information for the synthesis of proteins. So remember that proteins are the primary molecules that make all of the processes of life go. So it's through these proteins that DNA directs the development and life processes of organisms. So in this chapter, we're going to be examining the flow of genetic information, basically how we get from genotype, or the actual DNA sequences that an organism has, to the phenotype, or the outward projection, the physical manifestation of those DNA sequences. So we're going to be looking at the flow of genetic information. So the information content of genes is in the specific sequences of nucleotides. That's what we mean when we're talking about genotype. And the DNA inherited by an organism leads to specific traits by dictating the synthesis of proteins. So when we're talking about the traits, we're talking about the phenotype. So proteins are the links between genotype and phenotype. Proteins build phenotypes. So for example, a very small difference in the DNA sequence for a single protein makes the difference between these populations of old-field mice that are brown, that live in forests, and these lighter colored members of the same species of old-field mice that live on beaches and have lighter coats. Proteins are the link between the genotype, the differences in DNA sequences, and the particular phenotype, the traits that these organisms have. And we call this process gene expression, the process by which DNA directs protein synthesis, and it includes two stages, transcription and translation. So we'll talk about transcription in this web lecture and translation in the next one. But first, let's think a little bit about how we know what we know about what genes are and what they do, and how they specify the particular proteins that they specify. First of all, how is the fundamental relationship between genes and proteins discovered? How do we know that genes code for proteins? First, in 1902, a British physician named Archibald Garrod first suggested that genes dictate phenotypes through enzymes that catalyze specific chemical reactions. And so he thought that inherited diseases were caused by an inability to synthesize a particular enzyme. And so this was, the fact that they were inherited suggested that they have something to do with genes, and that enzymes were going to be responsible for whatever these symptoms are. So we know that cells synthesize and degrade molecules in a series of steps, and we call these metabolic pathways. And so scientists started to think about these metabolic pathways in terms of the genetic basis for them. And so this led to the one gene, one enzyme hypothesis. So George Beadle and Edward Tatum proposed discovering what genes do by basically knocking them out, making them defective. So their approach was to damage a gene and then observe the effect on the phenotype of having damaged that particular gene. And so what they did was they found mutants with defects in single genes so that they could determine what those genes do. And their results led to this one gene, one enzyme hypothesis, in which they proposed that each gene contains the information to make one single enzyme. And these ideas were later tested specifically in experiments by Serban Horowitz, who studied a three-step metabolic pathway that was well known at the time. And the end product of this pathway is arginine. We see the pathway down here at the bottom of the screen, starting with a precursor. Enzyme 1 catalyzes the conversion of the precursor to ornithine. Enzyme 2 catalyzes the conversion of ornithine to citrulline. And enzyme 3 catalyzes the final conversion of citrulline to arginine. And what they did was they performed a genetic screen by growing mold cells on medium that lacked arginine. And so we know that cells that died came from a colony that was missing an enzyme somewhere in this pathway. Somehow arginine wasn't being produced, and so if it wasn't provided in the medium, the cells couldn't grow. And so this led to more detailed experiments looking at each of these steps in the pathway. And so this is the experimental design. So the first step is to produce mutants that are unable to synthesize arginine. So we're going to produce a whole bunch of these mutants by exposing the mold to mutagens. And we're going to grow them on different types of media. So the first medium is going to be not supplemented with anything. So this is going to be minimal growth medium. The next medium is going to be supplemented only with ornithine. So that first step in the metabolic pathway, but no citrulline or arginine. The second one would be supplemented with citrulline only, but no ornithine or arginine. And then the final growth medium would be supplemented with arginine only, no ornithine or citrulline. If there are enzymes that catalyze each of these reactions from one precursor to the next in this pathway, we should be able to produce different kinds of mutants for each of these enzymes that catalyze those reactions. And so that's the prediction. And so what were the results that they found? They did find different kinds of mutants that could grow on different growth media. So we had some mutants that, if you provided ornithine only, could grow, but other mutants that could not grow on only ornithine. And then there were mutants that could grow on citrulline only, and the mutants that could grow with ornithine only could also grow with citrulline only. But there were also another group of mutants that could grow on citrulline only that could not grow on ornithine only. And then other groups with no growth at all. And then all of these arginine deficient mutants were able to grow on media that contained arginine. These results support the one gene, one enzyme hypothesis because each of these are going to have a defect in a particular gene, and we can see which enzyme each gene knocks out. So if we look at this figure from your book, we can see that in these mutants that could grow on the medium with ornithine, there was a defect in the enzyme that creates ornithine from the precursors. So if ornithine is provided in the growth medium, it can go through the rest of this pathway to produce arginine. And the second type of mutant that could grow with citrulline provided, there was a defect in the enzyme that converted ornithine to citrulline. So if that citrulline was provided in the growth medium, it could go ahead and produce the arginine and grow. And then in the third class of mutants, there was a defect in that third enzyme that converts citrulline to arginine. So that third class of mutant can only grow if the arginine itself is provided, the end product of this pathway. And so basically these experiments isolated these three distinct mutants that affected three distinct enzymes along this pathway. So their results supported this one gene, one enzyme hypothesis, but biologists realized that not all proteins are enzymes, and they finally understood that most genes contain the instructions for making proteins, and so that became the one gene, one protein hypothesis. But then, of course, there are many functional proteins that are made up of separate individual polypeptide units. Remember this idea of quaternary structure, where individual polypeptides come together to form a functional protein. Remember also that we only call it a protein when it has its correct functional shape. And so these proteins would only be proteins if each of those polypeptides are there. And so now we generally refer to this hypothesis as the one gene, one polypeptide hypothesis, but mostly we just refer to these polypeptides as proteins, which will eventually become either all of or part of a functional protein. So this led scientists to wonder, how does DNA code for proteins? What is that connection between the sequence of nucleotides and a sequence of amino acids? So Francis Crick proposed that the sequence of bases in DNA acted as a code. So DNA is an information storage molecule, and different combinations of bases are going to somehow specify the 20 different amino acids. And so he proposed that a particular stretch of DNA, otherwise known as a gene, specifies the amino acid sequence of one protein. The information in the sequence of DNA is not directly translated into the amino acid sequence of proteins. So we know that this doesn't happen directly from the DNA. So some other scientists, Jacob and Monod, suggested that maybe RNA links genes in the nucleus to protein synthesis in the cytoplasm. And finally, messenger RNA, also known as mRNA, was found to carry that information from DNA to the site of protein synthesis in the cytoplasm. So this ends up being the link between the genetic code in DNA and the production of the proteins. So the enzyme RNA polymerase synthesizes RNA using the DNA strand as a template. So it's going to do that complementary base pairing to copy the code by matching the complementary nucleotides. So this is that link again between the structure of DNA having these complementary base pairs and its function in being able to direct the synthesis of proteins by using another nucleic acid, RNA, that also will complementary base pair with it to transmit that code to the site of protein synthesis. And this leads us to the central dogma of biology. So the central dogma of molecular biology summarizes the flow of information in cells, which is that DNA is going to code for RNA, and RNA is going to carry the code for proteins. So the central dogma is basically DNA to RNA to proteins. That's the direction of the flow of information, and we can see that schematically here in this figure. DNA to mRNA. mRNA is going to leave the nucleus, if we're talking about a eukaryotic cell, and out to the cytoplasm, and there be transcribed using a ribosome into a protein. If we're going to define what is a gene, gene is a stretch of DNA, but not just any old section of DNA. Genes are specifically stretches of DNA that code for a protein. A stretch of DNA that codes for a protein, a single protein. The DNA sequence codes for the RNA sequence, and the RNA sequence codes for the sequence of amino acids in a protein. Before going on to see how that code was cracked, I want to mention a couple of exceptions that we know about now to the central dogma. First of all, many genes code for RNAs that actually don't function as mRNAs. They have their own separate functions, and they're not translated into proteins. So these RNAs perform important functions, for example, transfer RNA, which we'll see when we come to translation. There are microRNAs that have other functions in the cell. And so in this case, the information flow is just DNA to RNA without then leading to a protein in these cases. And there are also some cases in which information flows from RNA back to DNA, and this happens in certain viruses that contain an enzyme called reverse transcriptase. And so what reverse transcriptase does is it basically synthesizes DNA from an RNA template. So the virus carries the RNA, and then within the infected cell uses this reverse transcriptase enzyme to synthesize DNA from its own RNA, and that DNA can then be inserted into the genome of the host cell. So these viruses are known as retroviruses. HIV is a commonly known example of this kind of virus. And in this case, the information flow is RNA to DNA and no protein involved. So those are just a couple of exceptions that you should know about. So what are the roles of transcription and translation? What are these things? So transcription is the process of using a DNA template to make a complementary RNA. So this is basically just copying the information. So that's what transcribing is, right? If you think of the medieval scribes that were copying books, they were transcribing and just copying from one piece of paper to another. So transcription is basically just making a copy. Translation is the process of using the information in mRNA to synthesize proteins. So basically, you're changing from one language to another. You're switching from the language of the DNA code to the language of an amino acid sequence. And so we call this translation because it's the idea of changing languages. So DNA goes to mRNA through the process of transcription. So DNA is the information storage. Transcription takes this information to the information carrier, which is the mRNA. The mRNA is converted to proteins through the process of translation, translating the language of the DNA code or the nucleic acid code, the sequence of nucleotides, into the language of a sequence of amino acids. And so this information carrier, through the process of translation, is going to create the active cell machinery in the form of proteins. And then, of course, ribosomes are the sites of translation out in the cytoplasm. We've seen this at work when we had our tour of the cell. And so here's an image of an mRNA being translated by actually several ribosomes at the same time. So an mRNA molecule is generally translated simultaneously by several ribosomes in clusters known as polyribosomes. So here's a schematic. You can see one finished polypeptide being released from the ribosome, another one nearly finished, and these ribosomes are going to just keep attaching and moving along this mRNA until it's degraded. And here is a micrograph showing one of these polyribosomes attached to an mRNA strand. In prokaryotes, because there's not that separation of these processes of transcription and translation, translation of mRNA can begin before transcription has even finished. So here we have a prokaryotic cell. Remember, we've got the DNA, the chromosome, in this nucleoid region that's not separate from the cytosol, and we've got the mRNA being transcribed, and then immediately as it comes away from the DNA, it becomes attached to these ribosomes, which start translating the proteins. This enables the prokaryotic cell to produce those proteins very fast because it's going to happen immediately after each individual section of mRNA is transcribed. In a eukaryotic cell, the nuclear envelope separates the process of transcription from the process of translation. So in the case of eukaryotes, we have transcription completed within the nucleus before that mRNA is processed and then leaves the nucleus and encounters the ribosomes and becomes translated. So eukaryotic RNA transcripts are modified through RNA processing to yield the finished mRNA while they're still in the nucleus. Before they leave the nucleus to be translated, they're going to be processed, and we'll talk more about what exactly happens to the mRNA transcript. So what is the genetic code? The genetic code specifies how a sequence of nucleotides codes for a sequence of amino acids. It's basically the decoder ring that tells, or the language dictionary that tells you how to translate from the nucleotide code to the amino acid code. And a scientist by the name of Gamow predicted that each word would contain 3 bases, 3 nucleotide bases. And the reason for that prediction is just basically the math. So a 3-base code is the smallest that could specify 20 amino acids. So basically, if you have a 3-base code, there are 4 different nucleotides. That gives you 4 times 4 times 4 different combinations equals 64 different possible combinations. So we know that there are only 20, but if you have a code with just 1 nucleotide, that only gives you 4 different possible words you could spell or amino acids that you could code for, not enough. If it were just 2 bases, a 2-base code could specify a maximum of 4 times 4 equals 16 amino acids. We know we have 20, so we have to have more than that. The 3-base combination yielding 64 possible combinations is the smallest number that could give you the 20 amino acids, and it's the simplest solution So this was predicted to be most likely to be correct. And so a 3-base code is known as a triplet code. So what's the evidence that the nucleotide code is a triplet code? So a group of 3 bases that specifies a particular amino acid is called a codon. So Francis Crick and his colleague Brenner confirmed that codons are in fact triplets. How did they do that? They basically established a reading frame. So the sequence of codons of the gene could be destroyed by adding or subtracting either 1 or 2 bases. But you do not destroy the reading frame. You could still read the rest of that DNA code if you added or subtracted multiples of 3 bases. So don't worry about that if that's a little bit obscure. We're going to work on this in class a little bit to hopefully make it clearer. But this basically showed that these amino acids are specified by triplets of nucleotides. So another couple of scientists named Nirenberg and Leder determined which codon coded for which amino acid by making chains of specific codons and just assaying which amino acid was going to be bound to the ribosome with the known codons. So we now know for each of those 64 possible combinations what amino acid is coded for. So there's one start codon. This is going to be the very first codon in every transcript. And the start codon is AUG. It codes for the amino acid methionine, and it signals where protein synthesis is going to start, where you're going to start creating your protein. There are 3 stop codons, UGA, UAA, and UAG, that signal the end of the protein coding sequence. So that's where the protein is going to stop being synthesized. And the other 60 codons code for the particular amino acids. And so this is the translation guide. This is your secret decoder pen. So the way you read this is along the rows, you have the first nucleotide in the triplet. So in this first row, the first nucleotide in all of these is U. Second row, the first nucleotide is C in all of these columns, A all the way across, and G. In each column, you're specifying the second base. So as you go down the column, the second base is U all the way down, second base is C all the way down, second base is A all the way down, second base is G all the way down. And then within the boxes, you've got the third codon, U, C, A, and G. U, C, A, and G. Okay, so if you plot your triplet code on here, this shows you what amino acid you've coded for. So one thing you might notice is that we've got a lot of excess codons. We've got 60 codons that code for 20 amino acids. There's going to be duplication of codons that code for the same amino acid. So here we see 2 that specify phenylalanine, 2 that specify leucine, 4 more for leucine, and so there's repetition. The genetic code is redundant. All but 2 amino acids are encoded by more than one codon. Particular amino acids can be encoded by more than one codon, but it is unambiguous. So that means that each codon codes for one and only one amino acid. It never codes for more than one amino acid. So what do we mean when language is ambiguous? That means that it can mean more than one thing. So if we say something unambiguously, it only can mean one particular thing. So that's what we mean when we say the genetic code is unambiguous. A single codon can only mean one thing. It means one amino acid. But there can be more than one codon that codes for that amino acid. The genetic code is non-overlapping, so codons are read one at a time in sequence. And the genetic code is nearly universal, so all codons specify the same amino acids in all organisms with a few minor exceptions. So what does that tell us? This is very strong evidence that all of life is united in a single tree of life, all coming ultimately way back in evolutionary history from a single common ancestor that also used the same genetic code, the same sequence of nucleotides coding for the same amino acids. and the genetic code is conservative. So if several codons specify the same amino acid, it's usually the third nucleotide, the third base in the codon that varies. The first two bases are usually identical. And so if you look back at that chart with the genetic code, that's what you'll see. Generally, different codons that code for the same amino acid have the same two bases in the first two positions. And it's the third one that gives it what we call the wobble. You can mess around with that third codon and still get the same amino acid as a result. So we've got two strands of DNA. How do we know which one is going to be read? So one of the two DNA strands is called the template strand, and that's going to provide the template for ordering the sequence of complementary nucleotides in an RNA transcript. So that is the strand that's actually going to complementary base pair with the RNA to create that RNA transcript. So the template strand is always going to be the same strand for a given gene. So that doesn't mean that the other strand is never used to be transcribed. For some other gene, it might be the other strand, but it's always going to be the same template strand for a given gene. So the strand used as the template is determined by how the enzyme that transcribes the gene and what orientation it's going to attach to the DNA. That'll determine which of the two strands is going to be the one that's read, the template strand. And so this in turn is going to depend on the DNA sequences associated with the gene. So the particular DNA sequences that bind with the enzyme that's going to transcribe the gene, it's going to determine the orientation in which that enzyme binds. So during translation, the codons are read in the 5' to 3' direction. That means that the nucleotides, just as we saw in DNA synthesis, in the mRNA transcript, the nucleotides are going to be added to the 3' end of that growing strand. The non-template strand is called the coding strand, and we call it the coding strand because the nucleotides of this strand are identical to the codons in the mRNA transcript, except that T is present in the DNA in place of U in the RNA. So this is going to be what's the non-template strand is going to be the exact copy of the mRNA transcript, aside from the change from T to U. And each codon specifies the amino acid, one of 20, to be placed at the corresponding position along a polypeptide. So as that polypeptide grows, it's going to be a sequence of amino acids that corresponds to the sequence of codons in the mRNA transcript. So let's take a closer look now at how this transcription happens. So transcription is the DNA-directed synthesis of RNA, specifically a messenger RNA. So transcription, we can say, is the first stage in gene expression. It's the first thing that has to happen to be able to synthesize a protein. RNA synthesis is catalyzed by an enzyme called RNA polymerase, which is going to open up those hydrogen bonds between the base pairs in the DNA, so unzipping the two strands of the DNA molecule apart, and it's going to join together the complementary RNA nucleotides on the template strand. So the RNA is going to be complementary to the DNA template strand. It's going to complementary base pair with the DNA template strand. And unlike DNA polymerase, RNA polymerase doesn't require any kind of primer to get started. RNA synthesis follows the same base pairing rules as DNA, except that uracil substitutes for thymine. In the DNA sequence where RNA polymerase first attaches to begin transcribing the DNA is called the promoter region. In bacteria, the sequence signaling the end of transcription is called the terminator. It's going to be a little bit more complicated in eukaryotes, and we'll discuss that as we go. So that stretch of DNA between the promoter and the terminator, or the end sequence that we'll see in eukaryotes, is called a transcription unit. This is going to be the whole section of mRNA that you're going to have by the end of transcription. So here's a figure showing some of these concepts. So here we have the non-template coding strand of DNA being separated from the template strand by an RNA polymerase. Nucleotides are being added to this RNA strand at the 3' end, so it's being synthesized from the 5' to the 3' direction. Using the DNA as a template, it's going to add the complementary base pairs for the RNA and create these phosphodiester linkages, and they're going to be formed by the enzyme action of RNA polymerase. And this is going to happen after base pairing occurs. So first, the bases are going to be matched, and then once you have a match, then the sugar backbone is going to be linked up by the enzyme. So promoters signal the transcription start point and are usually going to be located several dozen nucleotide pairs upstream of the start point. So when we talk about upstream and downstream, downstream refers to the direction of transcription, so from the promoter down to the termination point, and the promoter is going to be upstream, so in the opposite direction from where the transcript is actually going to start to be made. And in eukaryotes, proteins called transcription factors are going to mediate the binding of RNA polymerase and the initiation of transcription. So here we see the RNA polymerase binding, unwinding the DNA to begin transcription at the start point a little bit downstream from the promoter. So in this figure we see the assembly of these transcription factors. So first we find transcription factors binding to the promoter region, and the binding of these transcription factors is going to facilitate the binding of the RNA polymerase enzyme. And once the assembly is complete of these transcription factors and the RNA polymerase II, we refer to this as a transcription initiation complex. So the combination of the bound transcription factors and the bound RNA polymerase is the transcription initiation complex. So the promoter region is known as a Tata box, and so there are lots of thymine and adenine nucleotides in this promoter region, and that's the signal for these transcription factors to bind there. The second step in transcription is the elongation of the RNA strand. So the first step was initiation, the second step is elongation. So as the RNA polymerase moves along the DNA, it's going to continue to unwind the double helix and separate the two strands about 10 to 20 bases at a time. So it's going to be this little moving bubble that comes along. And this is going to progress at a rate of about 40 nucleotides per second in eukaryotes, so a pretty fast process. And a gene can be transcribed simultaneously by several RNA polymerases. So you can imagine on this second one, this mRNA polymerase has gone by. Another one can now bind to that promoter region and start transcribing simultaneously. And nucleotides are added to only the 3' end of this growing mRNA transcript. After these nucleotides are added to the growing mRNA transcript, the RNA is going to separate from the DNA, allowing the two strands of DNA to come back together again, and so that is why we see this kind of small transcription bubble that kind of moves along, and this strand, instead of continuing to be complementary base paired with the DNA, becomes separated from it, and the DNA reforms the double helix. So as I mentioned before, the termination mechanisms are different in bacteria and eukaryotes. So in bacteria, the polymerase stops transcription at the end of the terminator region, and then the mRNA can be translated without further modification. In fact, as we saw earlier, it can start to be translated before it even is finished being transcribed. In eukaryotes, RNA polymerase II transcribes a sequence called the polyadenylation signal sequence. This is a very specific sequence of A and U nucleotides that signals the end of the transcript. So then the RNA transcript is released 10 to 35 nucleotides past where this signal is found. So the other way in which eukaryotic cells differ in their transcription process from prokaryotic cells is that eukaryotic cells are going to modify this messenger RNA within the nucleus before it's sent out of the nucleus to be translated. So enzymes in the eukaryotic nucleus modify this initial mRNA transcript, what we call a pre-mRNA, before those genetic messages are dispatched to the cytoplasm. So during RNA processing, both ends of the primary transcript are altered. And also, in most cases, certain interior sections of the molecule are cut out and the remaining parts spliced together. So first, let's look at what happens to the ends. So each end of a pre-mRNA is modified in a particular way. The 5' end receives a modified nucleotide 5' cap. So this is going to be, in this case, a modified guanine added to the 5' end. The 3' end is going to get what we call a poly A tail. So it's just going to be a whole series, can be from 50 to 250 adenine nucleotides added to that 3' end. And these modifications share several functions. First of all, they seem to facilitate the export of the mRNA to the cytoplasm, where the ribozymes are located. They also seem to protect the mRNA from hydrolytic enzymes, so keeps them from being degraded before they have a chance to be translated. And they also help the ribozymes attach to the 5' end to begin translation. Most eukaryotic genes and their RNA transcripts have long non-coding stretches of nucleotides that lie between the coding regions. So we see these long stretches. And these non-coding regions are called intervening sequences or introns. So this is the intron. The other regions are called exons because they're eventually expressed, usually translated into amino acid sequences. So this is very confusing nomenclature because the instinct is to think the introns are in, right? They're going to be in the translated protein, and exons exit or are out. But don't get confused with that. Remember, intron is for intervening. These are interruptions in between the sequences that are actually going to be translated. Exons, you can think of them as being expressed in the proteins. They're also the part that are going to exit the nucleus out into the cytoplasm to be translated into proteins. And the process of RNA splicing removes those introns before the transcript leaves the nucleus and then joins those exons together, splices them back together, creating an mRNA molecule with a continuous coding sequence. So the whole thing is going to be translated once it leaves the nucleus. So this is what the final mRNA that's going to be exported might look like. It's going to have its 5' cap. There's going to be a short untranslated region before the start codon, that AUG, which is the start of protein synthesis. This is all going to be coding until a final untranslated region beyond that stop codon, and then the poly A tail that helps to protect the mRNA. So in some cases, the splicing is carried out by a molecular machine called a spliceosome. So the spliceosome consists of a variety of proteins and several small RNAs. This is another example of those functional RNAs that are actually not translated into proteins, but just function as RNAs in the cell. Remember, we talked about those exceptions to the central dogma. This is one of them. And the small RNAs in these spliceosomes are going to recognize the beginnings and ends of these introns and help to recognize those splice sites. And they're also going to catalyze the splicing reaction. So rather than the proteins being the enzymes in this case, we have a case of ribozymes, so RNAs that function as enzymes. And we mentioned these back toward the beginning of the course when we were talking about the origin of life on Earth and the possibility of this RNA world where RNA serves both as the enzymes for catalyzing these metabolic reactions and also as the storehouse for the information. So ribozymes are catalytic RNA molecules that function as enzymes and can splice RNA. The discovery of these ribozymes rendered obsolete the belief that all biological catalysts were proteins. So this is very important in the thinking of the origins of life on Earth, because you can't have proteins before you have the information that codes for proteins. How could you have any kind of catalysis, any kind of enzyme activity before there were proteins? Well, this is how, using RNA as both the information storage and as the enzymes that are going to catalyze metabolic reactions. So 3 properties of RNA enable it to function as an enzyme. First of all, the fact that it's single-stranded means that it can form base pairs with other portions of the RNA strand, creating a complex 3-dimensional structure that's going to give it that tertiary structure similar to a protein that'll give it complex shapes. Also, some of the bases in RNA contain functional groups that can participate in catalysis. So when we talked about enzymes, about what's happening inside these active sites, that these functional groups are playing a role in lowering the activation energy. Some of the RNA bases can do a similar sort of thing with their functional groups. And also because RNA can hydrogen bond with other nucleic acid molecules, they can catalyze these reactions that involve other nucleic acids. So why do we have these introns? What are they doing there if they're just going to get spliced out and degraded away? We've discovered that some introns contain sequences that can help to regulate gene expression. When we talk about the regulation of gene expression, we're basically talking about controlling where, when, and how much of a protein product is going to be produced. So where it's produced, when it's produced, and how much of it is produced. So some of these introns might have sequences that help to modify the expression of the gene. And we've also found out that some genes can encode more than one kind of polypeptide, depending on which segments are treated as exons during splicing. So there can be what we call alternative RNA splicing at different points, depending on what polypeptide is being produced. So as a consequence of this, we found out that the number of different proteins an organism can produce is actually much greater than the number of genes, because a single gene can be spliced in different ways to create different polypeptides. It also turns out that proteins can often have a modular architecture, so that means that there can be regions of it that have distinct functions. So these distinct functional regions are known as domains. And it turns out that in many cases, different exons are going to code for the different functional domains in a protein. So for example, this exon 1 might code for this domain here, which is maybe the active site of an enzyme. Exon 2 over here might code for this domain that maybe attaches the protein to the cell membrane. Exon 3 might code for another functional domain that has some other function in the protein. So the importance of this has to do with the idea of exon shuffling, which occurs as a result of something called recombination in chromosomes, which we actually haven't talked about yet. We're going to come back and talk about chromosomes and how this recombination happens actually in BCOR011. But basically, this is a mechanism by which different functional groups can be switched onto different chromosomes to get different combinations of these functional groups to produce greater variation in the proteins. This creates variation for natural selection to work on. Most of these are going to be worse than the original protein, but every once in a while, this reshuffling of the exons will create a protein with a new function that works for the organism and is going to be favored by natural selection. So in this web lecture, We've talked about the first part of gene expression, the process by which DNA is transcribed into an mRNA transcript. We've talked about the genetic code that allows a sequence of nucleotides to indicate a particular sequence of amino acids. In the next web lecture, we're going to take this to the next step and see how that mRNA transcript is translated within the cytoplasm using ribozymes into the final protein product.